My name's Adrienne Jenner and I'm a PhD student, and I'm also a tutor here at UNSW for MATH1031 and MATH1131. I'm working in a field called oncolytic virotherapy, which is an area looking at treating cancer with viruses. The highlight of my career so far would be last year when I travelled to Mexico. I was luckily chosen to present at this conference in the south of Mexico in a place called Oaxaca. I was able to present my research to 40 of my peers and get their opinions and advice and their praise on how I was doing. And it was just wonderful to see how excited people were about this field of oncolytic virotherapy, and also to travel to this place in Mexico. It's just wonderful that math gets to take you all over the world. So in this field, what they're looking at doing is genetically engineering viruses so that when they're injected into the human body, they'll travel through the body and search for the tumour site. Then they'll start infecting the tumour cells. Once they're inside a tumour cell, they'll replicate many, many times within that cell and then burst out of the tumour cell, killing the tumour cell and then creating all these new viruses that can go on and infect nearby tumour cells. So the current virus we're working on is a PEG-modified adenovirus conjugated with Herceptin. Biologically, that means it's an adenovirus, which originates in our adenoids. We then coat it with a polymer called polyethyl glycol. This polymer shields it from immune detection so that once we inject the virus into the bloodstream, it can travel throughout the body undetected by our immune system and then arrive at the tumour site. Once it's at the tumour site, however, it doesn't necessarily know to infect or not infect the tumour cells. And that's why we conjugate the adenovirus with Herceptin. So Herceptin is a monoclonal antibody which binds to HER2, which is a protein overexpressed on breast cancer cells. So now we have a virus that can both hide from the immune system, but also target specifically breast cancer cells. In the field of medicine, we start off with a control, which in our case is just tumour growth without the effect of the virus. So what we did in the lab was they experimented on 6 mice and they measured the tumour volume over 60 days for these 6 mice. We then obtained the data for this. Now what we noticed with tumour growth is it's similar to an exponential function. When you think about it, tumour cells start proliferating very, very quickly and build up their population quite quickly, like an exponential function. So what we can use with maths now is an exponential growth to describe this. So we can also use the least squares fitting to fit our function to the data to obtain an approximation in a way to how the data is trending. We can then use this approximation to predict how the tumour is going to grow through time and further on. So what we have here is a plot of our tumour volume for each mice versus time. So every 2 days the mice's tumour volume was recorded, and that's represented by a blue star. There were 6 mice involved in the experiment. Once we obtained this data, we fit our exponential curve to the data using a least squares algorithm. From this we could see what the trend of the data was, and we wanted to use this to predict how the tumour volume would grow with further time. So now we've talked a little bit about how the tumour grows without the effect of the virus. If we wanted to include this virus into the model, we'd have to think about how they relate and how they rely on each other. So a virus will infect a tumour cell, it will kill that tumour cell and create all these new viruses that will then add to the virus population. So the populations change in relation to each other. So we can use a differential equation to describe how these populations will change and then hopefully predict how the treatment will work over a long time scale. The best memory of my undergraduate degree would be when I went on exchange to the UK. I was really lucky to get into a university called the University of Sheffield. I made all these amazing friends from around the world and since have been able to visit all these different countries to see them. And it was just incredible to go to the other side of the world and to study a subject that I love. And my passion for math really grew from going on exchange.